Hello everybody, my name is Carlos Coronel. I am a PhD student in Biophysics and Computational Biology at the Universidad de Valparaíso in Chile. I'm working in the Valparaíso Neural Dynamics Laboratory of the Centro Interdisciplinario de Neurociencia de Valparaíso in Chile. I will present some results of my current work in the laboratory, and the title of the presentation is Cholinergic Neuromodulation of Inhibitory Interneurons Facilitates Functional Integration in Whole Brain Models. The main idea of the work was to study how the neuromodulatory system managed the transition between different functional brain states from a fixed connector. That is, the brain hardware is fixed and the neuromodulatory system facilitates the transition between different patterns of brain activity. And first, as a motivation, if we think in a series of complex tasks, such as driving a car, playing piano, practice sport, or playing a chess game, we can divide those tasks in a set of subtasks. In the example of the car, when driving from our workplace to home, we need to incorporate visual and auditory stimuli. We need to perform motor movement to drive the car. Also, we need to use a working memory to map the roadway from work to home. In the brain, we have specialized brain areas to handle those particular features of the task. And this specialization of the brain function is known as segregation. But to produce a current behavioral outcome, we need to coordinate the activity of all those segregated brain areas, and this coordination of the brain activity is known as segregation. And in that line, two elements facilitates the switching from segregation to integration. One of the elements corresponds to the brain structural connectivity. It is hypothesized that the brain is placed between regular and random networks. That is, between networks that are completely segregated or completely integrated. But in short time scale, the network is fixed. An interesting question is how the brain manages the transition between different functional states based on a fixed connector. And the answer lies on the neuromodulatory system. In that line, hypothesis proposed by James McShane asks that two systems, the cholinergic system and the noradrenergic system, manage the transition between segregation and integration. Both systems modifies the features of the input to output function. That is a function that relate the incoming inputs to a population of neurons or neural masses to a certain output in the form of firing rate. It transfers the action potentials that receive a population of neurons in firing rate, in an output. In one hand, the cholinergic system modifies the response gain. The response gain controls the overall excitability of the system. Increasing the response gain increment the output of a population of neurons independently of the magnitude of the input. And on the other hand, we have the noradrenergic system, which modifies the filter gain. This filter gain is related to the slope of the input to output function, and the increment of the filter gain increase the responsivity of the populations to a strong stimulation, that is a stimuli that are relevant for the task, and decrease the responsivity of the population to weak or noisy stimuli that are not relevant for the task. And the hypothesis of Shine are that the functional integration is maximum at intermediate level of neuromodulation. That is, we need optimal levels of cholinergic and noradrenergic neuromodulation to maximize the functional integration. And for testing this hypothesis, we built a whole brain neural mass model based in the Janssen and Reed equations. The Janssen and Reed model is a model which can simulate EEG-like activity. That is, recordings related to electroencephalography. The model is presented in the figure A. In one brain region, or the cortical column, we have three populations of neurons. We have pyramidal neurons that are connected to excitatory and inhibitory interneurons, which provides excitatory and inhibitory feedback to the pyramidal population. The pyramidal neurons have projections to other cortical and subcortical areas and receive input from other brain areas scaled by a parameter alpha. We modify the model to include a connection between the inhibitory interneurons and the excitatory interneurons scaled by a parameter beta. This is a dysynaptic inhibitory connection. It's a novel add-on in the model that we include. And this is just for one node, and we connected the brain regions using a human structural connectivity matrix M parcellated in 90 brain areas, like prefrontal cortex, thalamus, hippocampus, among others. We used a structural connectivity matrix plus a speed matrix that is related to the propagation speed of action potentials between brain regions. The neuromodulation is presented in the figure C. For the cholinergic system, we modify two parameters, alpha and beta. Alpha scales the global excitability of the system, that is the global coupling between the brain regions, and beta scales this dysynaptic inhibitory connection between the inhibitory interneurons and the excitatory interneurons. Increasing beta attenuates the local excitatory feedback within the cortical column. So the effect of alpha is more global, and the effect of beta is localized is within the cortical column. Related to the noradrenergic system, we modify R0 that controls the slope of the input to output function of the pyramidal neurons. R0 is the filter gain of the pyramidal neurons. Finally, we have a whole brain neural mass model which can simulate EEG-like activity. One EEG-like signal for each one of the 90 brain regions that we considered in our parcellation. Also, the model can reproduce signals in two different timescales. In a faster timescale, we can simulate EEG-like signals with a resolution of milliseconds, and in a slowest timescale, we can reproduce signals of related to fMRI ball recordings that have a resolution of seconds. It's a slower timescale. Regarding the results, first we wanted to study the effect of the cholinergic system alone, so we fixed the filter gain R0 in 0.56. So we have the alpha beta parameter space that is related only to the cholinergic system. In the first row, in the row A, we have the phase synchronization and the mean oscillatory frequency omega, two measures calculated using DJ-like signals. And in the second row, we have the global efficiency, a measure of integration, and the modularity, a measure of segregation, both calculated using the functional connectivity matrices built using the Volt-like signals. And what we found that the neural coordination, that is the increasing phase synchronization and the increasing global efficiency, are maximum in an intermediate region of the parameter space. That is, we need optimal level of alpha and beta, and consequently, optimal level of cholinergic neuromodulation to promote the functional integration in a whole brain model. Also, the increase in the phase synchronization is accompanied by a decrease in the mean oscillatory frequency omega, a oscillatory frequency which falls within the theta range of the EEG spectrums, that is between 4 and 8 Hz, and this is reported in some EEG recordings when performing cognitive tasks, working memory tasks, tasks that require the use of math skills in other types of behavioral paradigms. Furthermore, the increase in the functional integration in the global efficiency is accompanied by a decrease in modularity. In fact, the modularity is higher near the boundary, which separates the unsynchronized region of activity in the black area and the synchronized region of activity in the blue area. So the message of this figure is that we require optimal level of cholinergic neuromodulation to maximize the functional integration at the whole, at the macro scale, that is in the whole brain. And in a more visual way, we can look at the Boltzmann-like signals and its respective functional connectivity matrices. We can change alpha and beta in the direction of this red line. Here we have the same subplot of the last figure, the global efficiency and the modularity. And from bottom to top, we increase alpha and beta in parallel. And what we found first for alpha 0 and beta 0, the network is nor segregated nor integrated. And if we increase alpha and beta little, the network becomes more integrated. Just in the middle with alpha 0.5 and beta 0.25, we observe the functional connectivity matrix that is very integrated, increasing alpha and beta further, produce a transition from integration to segregation. We can observe the segregation with the presence of two models with a high number of intramodular connections and a few number of intermodular connections. And if we increase alpha and beta at the most extreme case with alpha 1 and beta 0.5, the network, the functional connectivity matrix is not integrated nor segregated again. And the message is the same of the last figure. We need optimal level of cholinergic neuromodulation to maximize the functional integration in our model. And in summary, the dynamic gain that we include in our model promotes the functional integration in an intermediate region of the parameter space, and consequently, we need optimal level of cholinergic neuromodulation to maximize the functional integration. The results suggest a possible role of the synaptic inhibition that we include in the model, and is mediated by the inhibitory interneurons in shaping the whole brain dynamics. And this work is available online in bioRxiv. Here I put the link of the manuscript, and in the bottom the contact information, our emails, and the GitHub of laboratories in Valparaiso. So if you have some questions or if you want to contact us, write to the mails that I put in the last slide. And I hope that you enjoy the presentation. Thanks very much for your attention.